Welcome. We are here today to talk about our Center of Research Translation and how we're putting some complex data together on our patients with psoriasis. So I'm Kevin Cooper. I'm chair of the Department of Dermatology. Tom?
Tom McCormick, I'm the Director for Basic Research in the Department of Dermatology.
I'm Mark Cameron. I'm with the Department of Population and Quantitative Health Sciences, and I am the Director of the Applied Metaomics Core in the CORT.
I'm Jesse Preston in IT, and I'm the Manager of Enterprise Data Services.
Well, Jesse's worked with us before in pioneering how do we get data out of our electronic medical record for dermatologic quality measures, and now he's moved on to being able to help us integrate our omics data that Mark is working with, where we have hundreds of thousands of data points that are characterizing the patients molecularly, and then we're trying to figure out how to connect that over to the thousands of data points that are sitting in the electronic data from our electronic medical records, and that's what Tom is trying to do, is merge that together. So I'm going to start, Jesse, with you and say, so we've worked together for a number of years, and now on this CORT, it's really great to work with you again. Tell us a little bit how you get the data from various places, and how do you make this data able that we can then see it and merge it with very different kinds of data?
Sure. So we first go to the system itself, and we pull the data from that system in its raw format, and we stick it into our data warehouse in a staging area. Once it's in the staging area, it becomes somewhat of a temporal data lake, meaning that it's in its raw format. It's got many fields that would likely not be very usable for anybody wanting to mine the data or do analytics against it. And then it has some of those very relevant fields. Once we have it in there, then we go through a transformation process where we put it into our star schema of the enterprise data warehouse. But once we have it in there, it's often hard for a researcher or an analyst to navigate that star schema. So we take the data once it's been normalized and placed into that star schema, and we build data marts or data cubes. And that's a curated data set that particular researcher or that particular analyst is wanting to mine further. And it's got the fields that they need. It's somewhat trimmed down to the data set or data population that they want. And then if there were any calculated fields or functions that need to be applied to that data, we can do it at that point.
Great. And this data comes to us when we start to look at it. It's in a flat, almost like an Excel spreadsheet, for us to be able to work with. And then that's where Tom gets a hold of that and starts to try and put it into formats that are compatible with the kind of data that Mark is generating, which is coming from very different ways. And so we're trying to take these different kind of data sets and switch them so that they can be analyzed together. So Mark, I'm going to ask you, tell us a little bit about when you get the data from our patients with psoriasis. Well, you don't get the data, you get blood and cells and things from us, and so tell us a little bit some of the high dimensional analytics that you're using to try and look at that, and how many data points you're getting on a per patient basis.
Yeah. I mean, first you have to start with how much data is being collected from each patient, and the type of tests that we are running currently on the psoriasis patients for the CORT. The AMC, which is a component of the CORT, and the staff and the trainees that work with me, we have the type of machines that you can provide a blood sample loaded into the machine, and then you get tens of thousands of data points. And those data points relate to your genes and how they're expressed. And what the data approach is, which bioinformaticians go through and work on computers to find patterns in amongst the patients that have psoriasis and the various forms of that and the responses to the treatments that they have. So what we have at that point is a marriage between a lot of data that comes from technologies in terms of your genes and their function, and all of the data that Jesse was just talking about from the individual patients. And that has to be married and presented to the clinicians and the other parts of the court in a very visual and a really meaningful way. And only then can we start to find patterns that tell the difference between patient X and patient Y, or group of patient X and group of patient Ys, and provide that kind of precision information that may lead to new therapies or approaches to management of the disease. That is a very numbers intensive, computing intensive, and effort intensive endeavor. But it's produced the type of results after 2 years at this point in the program, where we have 100 patients that we're looking at, and we're trying to find and identifying these early patterns that we can then go forward with and target and make psoriasis treatment different.
So Tom, you've seen this data. You've seen Jesse's data in its raw form with all the millions of variables, and some of which are not numeric, some of which are numeric, which have, to be merged up will have to be changed into numeric data. You've seen the flow cytometry data. You've seen the transcriptomic data, these metaomic data. Give me your take on how do you see how this all goes together, and and trying to marry up things that are coming, identifying somebody from the molecular side, and trying to figure out how do we figure out the comorbidities of the unexpected human clinical side of this. How does this go?
Yeah, so I think Mark touched on it a little bit. It's in refinement, right? So the idea is we have this massive amount of data in tables that go on forever. And what we're really looking at is to make a more personalized approach for every psoriasis patient or group of psoriasis patients. So in order to do that, you need to know a lot about each one of those patients. So Jesse gives us the raw clinical data. Mark provides the transcriptomic and the gene data from that set. We have our own clinical data in the laboratory. And then when you look at all of these, you're just kind of glassy-eyed by how much data is there.
Yeah.
So our job now is to refine all of those data sets down to common elements that most patients would have that allow us to look at how they compare to each other. Data that mirrors the clinical side of what Mark see at the gene level, and then our own clinical data that we've done in the laboratory to add another component of that. So you want to make that flat file become more refined as we go, and we'll take 10 football fields worth of data and put it down into 2 football fields worth of data, and at the end, then we can begin to look for patterns using Mark's bioinformaticians to tell us about the analytics of how those patterns look alike or dissimilar, and then those groups of patients can begin to be identified. It really will work that way when we have all of this massive data, but trimming that data, which is the stage that we're in now, is a big job, and we're using medical students to do that. I'm coming to your office and saying what matters in a clinical state, so there's a lot of different moving parts right now, but that refinement is the process that we're engaging in now.
If I can pick up a point there, in terms of research data and the size of data sets that we've been working on, it used to be that a megabyte was a big number, and then a gigabyte worth of data was a big number. What Tom and Jesse are talking about, and the results that come off of our machinery and our bioinformatic analyses, we're not talking about terabytes. And that data sits on computers normally until it's dealt with in a workflow that can bring out the patterns from that massive information, but also get it across visually to the rest of the investigators within the court, and more importantly, get it out to the public so that it can be seen in print or in other forums like talks and posters, also in a meaningful way. And that's really what the modern field of systems biology and translation and big data is all about, is making use of our data, which is huge, terabytes worth of information, but also what other groups are doing out there, what's also been published in the literature, to assemble that information into large data sets that we can find patterns in. And if we can find the patterns and present them to each other and to the public, that's ultimately the goal of the court over its lifespan.
Yeah, this is great. And Jesse will remember when we did some early pulls from the derm EMR, just the derm EMR, it would kind of broke the system. It kind of, it ran for a really long time, and other things couldn't run on on the whole hospital. And so now we're talking about logs, more data coming in, and so trying to find the relationships. And one of the other variables as we're looking at the clinical data coming across, we're looking at what do we do about time. So time, stuff is happening in time. The stuff we're working with Mark on the AMC is a snapshot, but what's coming out of the electronic medical record is time. Things are happening over time with these patients, and so we're trying to figure out how do we quantitate that and put that into a number that can be merged in with the numbers that Mark is generating, that we're generating on the other side of the lab, and the CBC data as well. So it becomes quite complicated, and that's why we need really, really good programs. It's beyond what a human can think, and we need these high analytics that can do a little bit of thinking and finding the relationships between these values that will define new endotypes, new subsets of patients with psoriasis that have distinct outcomes. Precision medicine, really.
Yeah.
So anyway, I want to thank everybody for paying attention and listening to us, and you can see we're having a good time trying to figure this out, and enjoy working with you guys. Thank you.
Thank you.